This lesson is for Year 8 Science on Thursday the 10th of December, period 3. If you have any questions, Miss Cross will be available on Teams for answering them. Please use your book to complete any tasks and start by putting the date and title. You will find you need to pause this video to complete tasks as you work through the lesson. Please make sure you do that before moving on to the next slide with the question or continuing the video. So we are going to look at fertilisation and germination. Already this week you should have looked at the parts of the flower and pollination. Once you've done the date and title, here are 5 statements that have mistakes in them. The mistakes are highlighted in each sentence. Can you put 1 to 5 in your book, please, and correct the highlighted part? So correct the highlighted part. Please pause the video and do that now. Now you can check your answers. So we should have said the filament holds up the anther. The carpel contains the stigma, style and ovary. Those are the female parts of the flower. Wind-pollinated plants have brown or dull-coloured petals because there's no need for them to be colourful. Insect-pollinated plants will have a stigma covered in sticky pollen so that it can get stuck to the insects that come to it. And cross-pollination occurs between different plants. Before we continue, we need to recap some of what we've looked at in the first topic this year. So again, pause this video here and think about these questions. What process happens for reproduction? So there's a picture there to help you. Which cells does it involve? So again, the picture will help you here. And how does it happen? So the key word to include there is nucleus. Pause the video and write answers down to those 3 questions. So for reproduction to occur, we must have fertilisation. That process involves a sperm cell from the male and an egg cell from the female. For fertilisation to occur, the nuclei, so that's the nucleus of the egg cell and the nucleus of the sperm cell, must join or fuse in order to create a fertilised embryo. What we need to think about then is which parts of our plant that we've looked at might this involve for plants. So again, what sort of male and female parts have we learnt? Which ones are going to join in this process of fertilisation? So here are 3 simplified steps that show fertilisation in a plant. So we've got the first stage where we've got a pollen grain that has landed on the top of the stigma, and we've got something here that is starting to come down through the stigma to the style, through the style to the ovary. And when that pollen starts to move down, we get this pollen tube created. The nucleus of the pollen grain moves down towards this ovary. And once that pollen gets to the ovary, what is it going to join with? So we've got 3 stages. You need to use the parts highlighted on the 3 diagrams to create 3 simple sentences to describe what's happening at each stage. The questions you need to answer in your sentence are shown in the 3 stages below. So for stage 1, what have we got landing on the stigma? What is growing down the style? So the second diagram will help you with that. Stage 2, where does that need to reach and what moves down that pollen tube? And then finally for stage 3, what happens between the pollen nucleus and the ovule nucleus, so the ovule being the egg part, and what do we call this process? So we should have 3 sentences answering those questions. Please pause the video now to complete those. Hopefully we can now check those answers. Stage 1, the pollen grain, which is transferred from another plant if we've got cross-fertilisation, lands on that stigma. The pollen tube starts to grow down the style. In stage 2, the pollen tube will reach the ovary and the ovule that's in that ovary will have now be accessible. The pollen grain has got to move down that pollen tube and reach that ovary. Stage 3, the nucleus of the pollen grain and the nucleus of the ovule have to join for fertilisation to occur. So just like in animal reproduction that we've looked at, the two nuclei must join in order for that process of fertilisation to occur. When we've got fertilisation that has occurred, we will produce a seed. So this is a ovum or an ovule and a pollen grain that has met, and the nucleus has joined of each of those, and we end up with a seed. So here we've got a diagram of the seed, so you could draw that in your book. Okay, so we've got an outer layer, an inside part, and then we've got the green shoot that we can imagine as going to grow up through the ground. So have a look at these labels, pause the video, see if you can remember those three parts. So we've got a food store which contains starch. This is what the seed is going to use until it can start to produce its own food using photosynthesis. On the outside of that food store, we've got a seed coat. So this is a tough protective outer covering. And the embryo part is the young root shoot that will develop into an adult plant. So we can imagine this is going to start growing up through the ground and send roots down into the ground. Pause now, draw your seed and label it, please. So once we've got that seed is hopefully going to land somewhere that it can grow. But if it grows or not depends on certain conditions, and there are three things that seeds will need if they are going to grow. Using these diagrams, can you identify what the three substances are, A, B, and C, or conditions are that these seeds will need, and why do we need them? Okay, so each of these links from the diagram above to the diagram below. Pause now and predict what these three things are and why they are needed. Hopefully you had a go at that. So the first picture shows us a glass of water, and we hopefully know that plants definitely need water if they are going to grow. Now this water for the seed is required for the seed to swell and for the embryo to start growing. It's also going to need oxygen. Now, lots of you might be thinking, but plants take in carbon dioxide, and that is true. That's when they are doing photosynthesis. Before they can do photosynthesis, they need leaves, and our seed hasn't yet grown them. So before they can start doing their own photosynthesis, they need some oxygen, which they can get from the soil, in order to do respiration. And that respiration is the process where they release energy in order for them to germinate. So they need the oxygen in order to do respiration, to release energy, to germinate and start growing. The final condition they need is they need some warmth. So temperature helps speed up reactions. The higher the temperature, the faster the reaction can occur. So we need some warmth in order for that seed to germinate at a reasonable speed. So the warmth helps to speed up the reaction and germination. Quick questions then to answer in your book to make sure that you understand those three factors that we require and why. So answer those three questions quick as you can, summarising what we've just done there. Pause the video to do that now. Check your answers. So the three factors are water, oxygen and warmth. If we are missing one of those or one's limited, our seed might not grow, or it might grow much slower than others. The factor that provides or releases energy for respiration is the oxygen. So the oxygen combines with the glucose that's stored in that food store in order to release energy. And what happens to the speed of germination and growth in winter? Well, in winter it tends to be colder, so the germination and the growth would be slower because the chemical reactions would be slower, and therefore our reactions won't happen as quickly and we won't get our plant growing as quickly. Okay, so Monty Don is a very famous gardener. He presents Gardener's World, and he is upset. Only 2 of his seeds have germinated. The other 18 haven't done anything. He wants your help to work out the percentage of seeds that have germinated. So we can see here the percentage of successful germination is equal to the number of seeds that have germinated divided by the total number of seeds in the beaker multiplied by 100. Remember, percentage is out of 100. So can you use that information? There's a bar model there to help you because you've got to think about the total to work out the percentage of his seeds that germinated. Pause now and complete that. If we worked that out, then we should have identified that 2 plus 18 gives us 20 seeds total. 2 divided by 20 times 100 gives us 10% of his seeds that have germinated. So only 10% have germinated. Now, the sorts of factors that could have affected this, we've looked at the 3 factors required for germination. They are the water, the oxygen, and the warmth. It could be any one of those that has caused him to have problems here. So what we could do is we could do an investigation into one of those factors that affects the germination. So if we think about each year, tends to be spring that has nice weather. Lots of plants will germinate and grow during that period. And when we have a nice warm spring, we tend to get lots of plants growing and seeds germinating. What we're going to investigate, or what we could investigate, is whether or not the amount of water, so obviously if it's going outside, the amount of rainfall affects the amount of seeds that successfully germinate and grow. So what could that look like? So we could set up a number of beakers. So here we would be testing 7 beakers with different volumes of water. So we would set up the 7 beakers so that we could test 0 centimetres cubed of water that's been added, 1 centimetre cubed, 2 centimetres cubed, and so on. So we're adding different amounts of water. We would place our seeds and the water onto a piece of cotton wool. These seeds might be cress seeds, something that grows relatively quickly. We'd measure each volume using a measuring cylinder and pour it over the cotton wool pad in the beaker. Count out 20 cress seeds and sprinkle them over the pad, ensuring they are spaced out. Repeat that process, counting out 20 seeds for each beaker, and then cover the top of the beaker with some cling film and leave all the beakers on a windowsill for about 4 days. After that time, we would want to count the number of seeds that have germinated and record them for each volume of water we added. Pause the video here and identify what variable are we changing. Can you remember what we call that variable? Which variable are we observing or measuring? What do we call those variables? And what do we need to keep the same? What do we call those variables? Pause now, answer those questions using the information. So the variable we change is our independent variable. We can use the word chin to help us remember that. CH for change, IN for independent. The dependent variable is the one we measure or observe. We can use mode to help us remember that, measure, observe, dependent, and the control variables are the ones that we keep the same. So how do you think the amount of water will affect the germination of our seeds? Make a prediction. I predict that, and then more water, less water. One of those values of water will give us the most or maximum germination, and why do you think that? Hopefully we identified our variables from the previous slide. We are changing the volume, so volume of water. Our dependent variable, we are measuring the number that germinate, so we're counting how many germinate. And then control variables, we would want to make sure that we leave them for the same number of days, so we said about 4 days, have the same amount of light, so on that windowsill in the same position. We could also have identified here that we're going to put 20 seeds in each beaker. Okay, so our results. So obviously, unless you've got some cress seeds handy, you're not going to be able to do this one. But I have got some results to share with you here, so we'll use those results to have a look at how many germinated using each of those volumes of water. Unfortunately, some of the values are missing, so your task now is to complete the table for each of those covered values. If you need a little bit of extra help, I've put some extra help on here, so you can use the key for each of those shapes and hopefully use those to help you identify which is which. Pause now to complete that, please. So checking our answers, so they might just be off your screen. I'll make it a little bit smaller. So if we've got a mean of 0, we should find that our all of our dishes have 0. Hopefully we have identified our percentages. So to find our percentages, we needed to take our mean and divide it by 20, because we put 20 seeds total into each beaker, and then times that by 100 to get the percentage of seeds that did germinate from our practical. We had 3 of those to do. For this one here, we had to identify what our mean result was. So remember, when we're finding a mean, we're adding up our individual results and then dividing by the number in our set, which is 3 in this case. And then here, probably the trickiest one to do is identifying which number is missing. So we're doing the opposite of what we just did there. Instead of dividing by 3, we would need to times by 3, because that's the opposite. So we take our mean and times it by 3. Then we would want to take off each of our values for dish 2 and 3, and that should have left you with 10. Okay, so once we have collected that data, we could plot a graph of our results. Now, hopefully you have all recognised that we are going to need to plot a line graph here. We have got number data for both of our variables, so we are going to use a line graph. So we can see here that we've plotted a graph. You haven't got any graph paper likely at home, so we've plotted a graph of our results, and we've got the volume of water we've added on the x-axis and the percentage of successful germination on the y-axis. Remember the sorts of things when you are plotting graphs to ensure you do, so I've left those there in case anybody does have some graph paper and they want to have a go at plotting it. We could then use our graph to write a conclusion about our results. So again, pause the video now, have a go at using the graph shape to fill in this sentence in your book. Hopefully once you've got that done, we should see, so as the volume of water increases, so as we go along this x-axis, we're increasing the volume of water we're adding. Our line is going up, we're increasing the percentage of seeds that successfully germinate. But what we notice is when we get to our 8 centimetres cubed, the graph levels off. So the graph has stopped going up. The number that are germinating, the percentage that has germinated, is remaining the same. It's not going up or coming down. So have a think about what that tells you about the ideal amount of water that we could add to get maximum germination from our seeds. So the last thing to think about then, using our graph, is if we wanted to test another factor then that affects the germination of our seeds, we would want to use the optimum amount of water, so optimum being the water that is best for giving our maximum germination. We can use our graph to find it, so we would need to look at where we get maximum germination, but then think about what else we could consider. So would we just want to give 32 centimetres cubed to our seeds if it doesn't help germinate any more? Could we give 16 mil? Could we even give 8 mil? Why would giving lower amounts of water help our planet and our seeds? So it's about thinking about a little bit more than just giving the maximum. So hopefully here we would want to think about, we don't want to give any more than 8 centimetres cubed because any more would be a waste of water for the plant. So we didn't increase the amount of germination we got by adding 16 or 32, so there's no point. And so the second thing, it also helps the environment because the percentage of seeds germinating stays the same. To finish off then, so I'm just going to move these so that we can see them a little bit better. So you need to pick a minimum of 2 questions from each section. So section 1 is looking at the knowledge that we've looked at in these lessons so far. And then section 2 is looking at how science works and working scientifically. So I'm just going to move these down so that you can see them all. So in your book, to finish off, can you pick 2 questions from each section and answer them in your book to finish off the lesson? Move that over and that over, so you should be able to see all of those. So pause now, answer those questions. If you are unsure about anything that we've covered in this lesson, please ask Miss Cross. She is available on Teams during this Period 3 lesson. Thank you very much.